So we just finished up talking about the process of gene expression, but I mentioned a little bit earlier that we have all of this genetic information, and sometimes genetic information gets duplicated, and evolution can act on those duplicated regions. Um, Other times, changes get made to the DNA during the process of replication. Now, we haven't talked about that process, so I just have to take my word for it. But slight changes can occur in the DNA. And then I'll come back to this a little bit more in a few slides, but the environment can also cause changes in your DNA. So these changes in the DNA are really important in the evolutionary process, in allowing a population of organisms to change over time. Now, sometimes those changes are beneficial, but other times they're not. So, what causes changes in DNA sequence? So let's look at that. So a mutation is any alteration to a sequence of bases in the DNA. Generally, mutations are thought of as being detrimental to the organism. Now, this is because the organism already has a functional gene. And really, if you make any change to it, it's probably going to make it dysfunctional. Occasionally, mutations can be beneficial, and this is how we see evolution occurring. And we will, of course, get to that a little bit later, but basically, if a mutation is detrimental, the individual is probably not going to survive to pass on their genes. But if it's beneficial, they're probably going to survive and pass on that beneficial change. Quite often, mutations or changes to the DNA actually have no positive or negative effect. They're completely neutral. So if they occur in the junk DNA region or other non-coding regions of the DNA, they actually probably aren't going to have any effect. Now, something else I wanted to point out, we looked at the genetic code a few minutes ago, and we saw that there are multiple codons that code for the same amino acid. You can also have a change that changes from one amino acid to a sequence that codes for the same amino acid. So that's another way you can have a neutral mutation. But no matter if they're good or bad, mutations are essential to evolution. They are the only source of new genetic information in an organism. And so they are one of the key ways that evolution can act on a DNA sequence. So when a change occurs in a coding sequence, you end up with a different protein. Sometimes you end up with no protein at all or a dysfunctional protein. And that leads to an alteration in phenotype. So here we have a mutation in the fruit fly eye that is produced by an altered gene. So in this gene alteration from this normal to the mutant fly, the fly essentially loses its entire eye. So this is called the eyeless gene, and when the mutation occurs in an eyeless gene, you end up with a fly with no eye. So there are different types of mutations, and we're going to talk about some of those types in just a minute. The location of each of those types of mutations is really important too. So mutations that occur in non-sex cells are important for the life of the individual, but are not passed on to offspring. So, for example, if you have a mutation that occurs in a lung cell or a skin cell, that can potentially cause cancer in those regions or other types of medical problems, but they're not going to be passed on to the individual. Passed on to the individual's offspring. However, mutation that occurs in sex cells are potentially passed on to the next generation. So our sex cells are going to be our sperm or our eggs, and these are called gametes. So when a mutation occurs in a gamete, it's not going to affect the individual that possesses that gamete at the moment, but they can potentially be passed on to offspring. So these are the ones that are going to be the focus for us because we are worried about propagating life and passing on information to the next generation. So whether or not they occur in non-sex cells or in gametes, there are two major types of changes to the DNA that can occur. One is a point mutation, which is a change in a base pair. The other is a chromosomal aberration. So these are changes in the physical characteristics of a chromosome. So point mutations are changes to one or more nucleotides. Generally we think of this as replacing one with another nucleotide, but it can also be an insertion or deletion of one or more nucleotides. So we have our image here, normal DNA. So here's our DNA coding, our DNA template strand. Here is our messenger RNA that we've made with that template strand. And then down here is our amino acid sequence that we've begun to make from this mRNA. Remember, we've got our start codon here with our start methionine there. So let's look at these different types of point mutations and see what happens. So the first is a substitution. So this can be any change. It can be a adenine to a thymine. It could be a cytosine to a guanine. It can be a purine to purine, or pyrimidine to pyrimidine, or a purine to pyrimidine. Any combination that changes the base pair in the sequence. So when that happens, if we have a change in our DNA, let's say we go from a T to an A, so that T is changed to an A. A is changed to a T. That changes the messenger RNA sequence, which then changes the amino acid that is inserted. So originally we had methionine followed by cysteine. In our mutated protein, we now have methionine followed by serine. Now this is potentially going to change the physical characteristics of the protein, which is then going to change the function of the protein. So substitutions are very important because you can change the functionality of our protein. So something like that is potentially going to cause a mutation like the eyeless gene in the previous slide, where now you have a protein that doesn't function at all in the organism. You can also have a nucleotide insertion. So a nucleotide insertion is going to cause something called a frame shift. In a frame shift mutation, you're going to insert a base pair, and that is going to shift your 3-letter codon code. So here we've got, in the original, we've got TAC, ACG, TCG. But down here, whoop, we inserted an extra C, and now instead of being TCG, our codon is CTC. Now that affects everything after it. It changes every triplet following the insertion. Now, not only have we changed the amino acid at the site of the change, but we've changed everything after it. So the substitution only changed one base pair. It left all the other only changed one amino acid, which left all the others. In an insertion, we've now changed everything afterwards. This is very likely going to produce a non-functional protein, which could potentially be detrimental to the organism. We can also have the same sort of situation and remove a base pair. So when we do that, we get a frame shift back a little bit. So instead of TCG, we now have CGC. So you see we've lost this T, and we have changed the triplet code for everything following. So this deletion has caused a different set of amino acids to appear in our mutated protein. These insertions and deletions are really detrimental because they are changing everything following. The nucleotide substitution severity is just going to depend on the change that happens. If you insert a similar amino acid, you might not have as much of an effect. If you insert a different one, a very different one chemically, you can have a major change in protein function. You can also introduce a stop codon, which would terminate the protein prematurely, and that same thing could appear in an insertion and deletion as well. So if you introduce a change that makes a stop codon, you are obviously truncating your protein and rendering the entire protein dysfunctional. So we're going to do an assignment in class where we're actually going to look at these different types of mutations and see what they do to the sequence in question. So our second type of mutation is a chromosomal aberration. So this is a change in the physical characteristics of the chromosome itself. So there are several ways that this can occur. You can get a gene deletion or a region deletion, where say a breakage occurs, and in an attempt to repair the break, the cell loses a chunk of DNA. This can also occur from an exchange in cell division called crossing over, where the chromosomes that originally came from your mom and dad don't correctly exchange genetic information at a particular point, and some information can be lost. You can also have a gene relocation. So sometimes genes do this really cool and weird thing where they pop out from where they are and they reattach themselves somewhere else. Now, this is obviously going to affect the chromosome as a whole. If the gene is lost, if sections of the gene are lost, the whole thing isn't moved together. You can lose gene function. You can also change how that gene is regulated or expressed. If it's in a different region, it might be regulated or expressed differently. Then we can have gene duplication. So some of the processes that I talked about with deletion, this unequal exchange or repair problems, can actually cause a section of DNA to be duplicated. Now, all of these can be really important in evolution, but it's that gene duplication that actually allows those duplicated genes to potentially take on a new function. It's also how we get those pseudogenes that we talked about in the first video, where if there's another copy that's producing a functional protein, maybe that one's not actually maintained. And so there comes a point where it's not really even used in the genome. So there are several chief causes of mutation. One is spontaneous mutation. Now, this is the one that I was talking about that comes from errors in DNA replication. So the process of DNA replication is actually really good. It has to be, because it has to copy a lot of information in a short time, and it has to make sure that it maintains the genetic information in the cell without changing it. Because if you're a skin cell making a new skin cell, you need the correct copy of information. If you are an oocyte, that is the cell that is going to turn into an egg, for reproductive purposes, you want to make sure that you pass on the correct information to the next generation. So the process of DNA replication actually incorporates proofreading capabilities, which is really cool, I think. But it's not perfect. It's just like sometimes you might proofread a paper so many times and you still turn it in with a typo. It happens. It happens in the DNA. Sometimes it occurs in those non-coding regions and doesn't affect the individual. Other times it does occur in a coding region and is potentially a problem. Environmental factors also cause mutation. So radiation-induced mutation are important. So we have UV radiation, X-ray radiation, things like that that can all cause changes to the DNA. And this happens because those radiation waves actually interfere with the bonds that are holding the DNA together. And so you can get changes in the structure of DNA based on changes in those bonds between the base pairs. And this can cause problems in DNA replication and gene expression, because the cell won't know what information it needs to be reading, or it won't recognize the information at all. You can also have chemical-induced mutations. So these are things like mutations that are produced from smoking or asbestos exposure, things like that. Same type of thing. The chemicals can actually interfere with your DNA and change the information, cause a mutation of some sort. So faulty genes are really important in a variety of illnesses. Now, this might be something like cancer, where your exposure to sunlight or smoking causes a change in a particular cell. So obviously those are major illnesses that are due to mutations. But there are some that are less, I would say, quote unquote, important. Some that are not necessarily deadly, but really inconvenient. And they are almost always due to a lack of functioning protein at the end of the pathway that they are part of. So we have this process of gene expression. So we go from DNA to messenger RNA to a functional protein. And that protein performs a particular job in the cell. Let's say that protein's job is to break down lactose in milk. If you acquire a mutation in that gene, your DNA is no longer coding for the correct protein or enzyme that's going to break down that lactose. Or you may have been born with a dysfunctional gene so that your body doesn't produce lactase, which is the enzyme that breaks down lactose. So let's say this is our functioning enzyme. This is our functioning enzyme, and it is going to recognize our lactose molecule, binds it, breaks it down, you drink your glass of milk and go on your merry way. Let's say you have a non-functioning version of that gene, and so you cannot break down lactose. That mutation in the DNA is going to produce a slightly different shaped protein or enzyme that no longer recognizes that lactose. So it can't break it down, and it can cause problems. Something that I found really interesting, in the United States, this lactase gene is actually really common, but in the rest of the world, it's actually not very common. Being able to digest milk past being a baby is actually not the norm. This was actually the ability to break down lactase actually arose in a variety of different cultures that have been historically dependent on dairy animals, so cows or goats, where it was actually advantageous to maintain this functioning lactase molecule past breastfeeding, past weaning. And so a lot of individuals of Asian descent actually don't have this, are actually lactose intolerant. And we call it in the United States intolerant, like it's the weird version or the bad version, when actually internationally lactose intolerance is kind of the normal. There are some other problems that can arise, some other examples of faulty genes. Your book goes through something called fast flushing, which is an inability to produce something called alcohol dehydrogenase, which breaks down alcohol in your system. Albinism is another form of a faulty gene. So in albinism, the body does not produce pigment. So the individual appears to have no pigment. Their skin is really light. Their eyes are often red in color. And then we already talked about Tay-Sachs and cystic fibrosis in chapter 4. So those are all good examples of faulty genes that can lead to a sickness.